All right, time to go teach. Hello and welcome to this next episode of Tutors, continuing biochemistry and looking at structures in our polypeptides. So last time we talked about how amino acids were the monomers of a polypeptide, and now we're going to look at how those different monomers can interact with each other to give the whole polypeptide a specific structure and hopefully a specific function, so that way it can be called a protein. So let's get going. Here's how it works out. Targets of the day, really simple. Describe each level of structure in a polypeptide. That's the whole thing. Now, let's begin. There are 4 different levels of structure for a polypeptide chain. There's primary, secondary, tertiary, and quaternary structure. So we're just going to go through each of these one at a time. One thing I want us to recognize, though, is that it's not like any of these things exist without the other ones at the same time. Specifically, primary, secondary, and tertiary structure, they're found in every single polypeptide chain. Quaternary structure isn't found in every polypeptide chain. It's a little bit different. But the first 3, every single polypeptide chain will always have primary, secondary, tertiary structure. So, some things to think about when we're thinking about structure. Something just to, mull over. When we're talking about paintings, when you're looking at a painting, you can look at the whole painting, but you don't have to just look at the whole painting. You can actually analyze what was going on in the artist's mind when they decided to make that painting. What type of brush did they use? What type of paints did they use? What colors? How did they end up mixing different techniques together? That all gives us the painting that they end up making. Movies, it has multiple parts. You can analyze the script, you can analyze the acting, you can talk about the directing or the cinematography. The key is it all comes together to make that specific movie. Or writing, you have the order of the words, you have the order of the letters that make up those words. So you have the way that the sentences are structured, and you have the way that the entire story is built and fleshed out. And all of that, those are different levels of structure for each of these different pieces of art. For a polypeptide, they also have levels of structure that build upon each other, and they don't exist in the absence of each other, but we can focus on one level of structure over a different level of structure. To do this, we're going to simplify how the amino acid looks. So instead of drawing the ball and stick model, what I'm going to do is just have this simple structure right here, and now we have a chain. This is our polypeptide chain, and that brings us to our first level of structure, which is primary structure. And that is just the order of the amino acids. It is very simply which amino acids are there and in what order they come in. So in this case, our primary structure would be methionine, cysteine, glycine, arginine, tryptophan, cysteine, valine, etc. So in our simple polypeptide chain here, that is the primary structure, the order of those amino acids. The primary structure is going to determine the next level of structure, the secondary structure. So the secondary structure is based upon the primary structure, which makes sense because primary would be the first level, secondary would be the second level. And so the primary structure is going to have a large impact upon the rest of our polypeptide chain. In fact, if we were to change one of these amino acids out and replace it with something else, that could have massive effects on if our polypeptide chain could function or if it couldn't. So that could change it from being a protein to just a polypeptide. So that was why primary structure is really important. Next, secondary structure have multiple ways in which they can actually be expressed. So the secondary structures, that is due to hydrogen bonding within the polypeptide chain but using the backbone. That's that nitrogen, carbon, carbon backbone from our amine group, our central carbon, and then our carboxyl group. That part can have hydrogen bonding take place between the amine group of one amino acid and the carboxyl group of a different amino acid. Because the amine group, that hydrogen attached to the amine group is going to be slightly positive, and that oxygen that's still attached to our carboxyl group, that will be slightly negative. So they can hydrogen bond together. And thus, it can form some interesting structures. In this case, it's forming what's called an alpha helix. An alpha helix is when we end up with a spiraling structure. So it spirals around like a coil. Okay, sort of like a slinky or a spring. That is an alpha helix, and that's due to hydrogen bonding between amino acids that are approximately 4 amino acids separated from each other. As long as our polypeptide chain hydrogen bonds about 1 every 4 amino acids, it's going to make this structure, which would be an alpha helix. Just a little spiral. Or another thing that could happen is if we were to add a couple new amino acids, we just added histidine and lysine right there. What else could happen is this something called a beta pleated sheet. And that's if the hydrogen bonding between the backbone happens at further intervals. These could be hundreds of amino acids apart, or they could be, say, 10 or 7 amino acids apart. It varies, but the key is that now we're going to end up with the chain going back and forth and back and forth and back and forth, weaving, in a sense, around hydrogen bonding between itself to make basically a zigzag pattern, which would be our beta pleated sheet. That is the other structure that can exist in the secondary level. So primary level, that's the order of amino acids. Secondary level can form either an alpha helix or a beta pleated sheet, but both are due to hydrogen bonding, which exists between the backbones of our different amino acids. So that is our secondary level of structure. Tertiary is now where we're going to talk about the R groups of our amino acids interacting. We've already formed our alpha helices, we've already formed beta pleated sheets, and now those structures are going to interact with each other and make a specific shape for our polypeptide chain based upon the interactions of the R groups that are now sticking out. So we can have an alpha helix that is just going to spiral down. And coming off of that alpha helix, well, could be another alpha helix, or we could have a beta sheet. So in this case, we're going to have a beta sheet come off of that alpha helix, and see right here how the beta sheet is arranged with the alpha helix is because of hydrogen bonding, or polar to polar interactions, or non-polar interactions, or maybe even a disulfide bridge at that location. Because of those R groups interacting, that's going to cause a specific shape. Here we have another alpha helix that came off of that and clearly interacting with these. So we have a bunch of interactions between these different secondary structures that are based upon the R groups of the amino acids interacting. That gives us our tertiary structure, and now this chain is folded up into a specific shape. They get their shape based upon these different levels of structure. First, the primary level of structure, which is based upon just the order of amino acids. Then we have the secondary levels of structure, which is going to provide us with either alpha helices or beta sheets. And third, the alpha helices and the beta sheets are going to interact with each other based upon the R groups of the amino acids that are present, and they are going to form a specific overall structure for our polypeptide. Again, if it functions, it's a protein. If it doesn't function, that's still just a polypeptide chain. But there's another level of structure that can be included. And that next level can maybe take this structure and turn it into a protein. And that is quaternary structure. Quaternary structure means that another polypeptide chains, R groups, are going to interact with our original polypeptide chain. And as long as we have multiple polypeptide chains interacting, we have what's called quaternary structure. If, because of these multiple polypeptide chains coming together, they now can perform a specific function, then they're a protein. And so many proteins have quaternary structure where originally that polypeptide chain was just a polypeptide chain, but now that it's interacting with another polypeptide chain, or maybe two other polypeptide chains, or 3 or 4 or 5, as long as it's interacting with other polypeptide chains to form a single functional unit, that would get us to quaternary structure, and that would also make it a protein. So now we have a full unit that is a protein. Each of the single polypeptide chains would be called a subunit at this moment, because the entire protein would be referred to as a single unit. So they are the subunits that make up that unit. And those are the 4 different levels of structure that you can find with polypeptides. You can have primary, secondary, tertiary, and quaternary structure. The first 3 always exist. Even if you only have a few amino acids in line, they will have primary, secondary, and tertiary structure. But to have quaternary structure, you must have multiple polypeptides interacting. So in summary of all of that, straightforward, we got all polypeptide chains. They can be studied at those different levels. Sometimes a scientist is going to be just wanting to study what different alpha helices or what different beta sheets are present. Sometimes a scientist is going to want to see how do those alpha helices and beta pleated sheets, how do they interact with each other, thus forming the specific shape of that polypeptide chain. That would be the tertiary structure. Or sometimes you want to know the exact ingredients that make up that polypeptide chain, the exact order of amino acids, and that would be the primary structure. So it all depends on what level you want to study your protein or your polypeptide chain at. Next, all polypeptide chains have, again, that primary, secondary, and tertiary structure. They have to. There's no way of avoiding it. There's going to be an order of amino acids. The backbones are going to interact, and the R groups are going to interact in some way. And some have to have another, a 4th layer of structure, which would be another polypeptide chain interacting with them. So that's how we analyze our proteins, from those different levels of structure, so we can determine exactly what they're made of and how they're put together. So we can understand the structure, which helps us understand the function and how they can function. So that's the 4 levels of structure that a polypeptide chain or a protein could have: primary, secondary, tertiary, or quaternary structure. And we can analyze our proteins or our polypeptide chains at any one of these levels to fully understand what's going on at a deeper level. So that is it. Until next time, be awesome, stay awesome.